Hello, today I'm going to talk to you about fast and efficient mixed effects algorithm for longitudinal data analysis. This is a new method that we have developed called FEMA. I'll introduce the idea behind FEMA and talk to you about a few of the applications. I do not have a conflict of interest, and these are my funding acknowledgements. In general, we know that we need large-scale sample sizes for achieving accurate effect size estimation. And typically speaking, longitudinal data is necessary for estimation of how various effect sizes change over time. However, as we grow sample sizes and as longitudinal data comes in, so do various kinds of complexities. For example, these are some of the data sets that we are very familiar with. For example, the Human Connectome Project, the ABCD study, the UK Biobank. Otherwise, we also have data sets from let's say the Norwegian pregnancy cohort called MOBA. We have the UK Waste Protect study. Various Nordic countries have health registries and patient records which are across the entire population. All of these data sets have various complexities, like they are longitudinal in nature, they may contain related subjects, and so forth. Let's consider the simple example of time and a particular y variable, in this particular case weight. This is how the data looks when we account for the fact that these are longitudinal data points. So a simple linear model would be that weight is a function of the intercept and the time, or week in this particular case. However, this model assumes that the error term epsilon is uncorrelated across observations. Additionally, notice the fact that there is substantial variation across these individuals, so it might not be a good idea to fit a simple single intercept model. Also, the fact that these are repeated observations needs to be accounted for. So, one could fit an alternate model, in which case we can have an intercept for every single observation. In this particular case, 48 observations, or 48 indicator variables, and then the week and the error term. But this is not really a good idea because there are too many parameters and this model cannot generalize to other observations. The same model could be reformulated in the context of a mixed model where we have the fixed effects, in this particular case the intercept and the week, and the random effect, which is characterized by the VS term here. This will have a single parameter, the sigma square S, which indicates the variance across the subjects. So in this particular case, we have two fixed effects and one random effect. And what we're trying to say in general is that some of the variance in the data is being explained by various grouping or nested structure that is present within the data set. And the example of such structures could be repeated observations, it could be relatedness among sample, it could be kids going to the same school, other kinds of factors like shared environment, and so forth. An alternate way to think about it is the weighted average between two extremes. One extreme is the first model which I showed, in which case we don't account for any level or grouping structure in the data, and the other being where you have every single observation was shown as a separate indicator variable. So mixed models are in an approximate weighted average between these two extremes. Well, in general, mixed models are fairly powerful and versatile approaches, and they can model things like longitudinal data and other kinds of known sample variances. But you might be wondering, why should we bother with mixed models? Well, let's look at this particular example. In this case, we have the price on the x-axis and the sales on the y-axis. And if we fit a simple linear regression model, this is how the fit looks. However, if you account for the fact that these are longitudinal observation or the prices and sales of the same commodities over time, and then fit the regression model, this is how the model would look. So the key thing that I want to highlight here is that if we fail to account for the clustering of various kinds of structure which are present in the dataset, we could find very different interpretations. Other reasons to consider doing mixed models is that it improves statistical power, it provides a better control over type 1 error, and you can handle things like unbalanced designs very easily. So in general, when you consider applying mixed models for large-scale dataset, where large scale could be in both in terms of the number of observations or in terms of dimensionality of the dataset, it can take a long time to run. And FEMA is a fast and efficient solution that can scale very well to both large number of variables and to a large number of samples. Well, you might wonder, how long does it really take? Consider the example of a standard T1 weighted brain, which has about 550,000 or so voxels. If this model fitting for a single voxel takes about half a second, the analysis would take about 3 days to run. And if the same model took about 30 seconds, we'll be talking something like 190 days. And 30 seconds is just a simple linear mixed model. Sometimes fitting these can take several hours. This is an example of how long a standard model fitting for mixed models would take, which is plotted in orange as a function of number of observations. And the time taken by FEMA is plotted down here in green. We even tried to fit this using a parallel computing approach, and you can still see that FEMA outperforms even the parallel computing case. This is another example of how the timing scales with the function of number of y variables. And we've plotted here about 5,000 y variables from 100 to 5,000. And you can see that the standard linear mixed effects model would take around, for 5,000 variables, it would take about 5,000 or so seconds, while FEMA, the timings are about 5 to 10 seconds. Generally, FEMA achieves this kind of computational efficiency because of a variety of reasons, but the key factor being that we have a methods of moment-based estimator rather than a maximum likelihood estimator, and the fact that everything is written in a highly scalable vectorized fashion. Apart from that, we also employ a very efficient binning strategy. What that just means is, if the random effects look very similar across brain voxels or vertices, we tend to estimate the total variance together. The key thing that I want to highlight here is that FEMA achieves this kind of computational efficiency without compromising estimation accuracy. In this particular example from the ABCD study, we fit an ROI level cortical thickness model across 6,314 subjects, and this was repeated measures, so there were 2 time points. This is how the model estimates look for each of the various ROIs on the left and right hemisphere. The points in green are the estimates and the confidence interval from FEMA, while the one in orange are the estimates and the uncertainty estimates from standard mixed effects modeling. And you can see there is absolutely no difference between the two. So what are the things that we can do with FEMA? The first thing we can do is heritability estimation. Heritability is the proportion of variance explained in a particular y variable or phenotype by the genetics. So in this particular example, we looked at the cognitive measures from the NIH toolbox in the ABCD study. Again, we looked at 2 time points. These are the cognitive measures and the tasks that we looked at. And in this particular analysis, we found that different phenotypes had varying levels of heritability. Heritability estimate here is shown by the A factor here, which is additive genetic effect. The other thing that you could do is, of course, as we've already discussed, the accurate estimation of various variables of interest. For example, in this particular analysis, we looked at the ABCD dataset across the 2 time points and asked the question, how does cortical thickness change as a function of aging? And the map here shows a vertex-wise analysis where we looked at the change in age and how does that affect the cortical thickness at each of the vertices. Similarly, in this particular analysis, we looked at how the resting state functional connectivity both within and across different ROI oscillations change as a function of change in age. The other thing that we could do within FEMA is, of course, to look at the random effects themselves. This is examination of 3 different random effects on cortical thickness in the ABCD study. The first one here is the additive genetic effect, or how related, genetically related are the subjects within the ABCD study. That's akin to the heritability estimate. C is the common environment, and S is the fact that these are repeated observation. Same analysis but using surface area, and the same analysis using circle depth as the Y variable. Here, for example, the subject effect is extremely large. These were some of the examples of what FEMA can do. Now we have extended FEMA to perform large-scale GWAS analysis. Interestingly, what we are also looking at these days is how can we use FEMA to model the longitudinal phenotype and discover how do different SNPs drive the change in phenotype. Or in other words, what we're talking about is a simple SNP intertime interaction, saying given a longitudinal phenotype, how does the effect of the SNPs change over time for this particular phenotype? This is just an example to demonstrate that the cross-sectional GWAS works. This is just a QQ plot which shows that when there is no genetic effect, we get a fairly flat line as expected. And this is an example of various simulated ground truth and the estimated beta coefficients across different SNPs, and everything looks as expected. We have also applied FEMA to various real phenotype, for example, placenta weight from the Nordic mobile dataset, and to gray matter volumes in the UK Biobank. To summarize, the key things that I want you to remember from this discussion is that datasets have various kinds of complexities which we should account for, and the fact that FEMA can account for these random effects in a very fast and efficient manner. Upcoming releases of FEMA will allow you to model how the effect of the interaction between genotype and age, and also genotype, age, and sex as smooth nonlinear interactions. I want to thank all my friends and collaborators who are key people behind FEMA and FEMA-GWAS, including Anders, Tom Nichols, Ulla Andreasen, and various people at Normand and UCSD. And thank you for listening.